Hello and welcome everybody to today's Drug Hunter Flash Talk. I'm your host, Dennis Koester, and we will be talking about molecular storytelling with Martin Stahl today. Good evening, Martin.
Hey, Dennis. Pleased to be
Yeah,
here.
good to have you here today. Without further ado, I want to introduce our guest, Martin. So Martin Stahl is currently the CSO of Zenith Therapeutics. That's a company pioneering novel molecular glue degrader approaches. Prior to his current role, he's been a CSO at LifeMine Therapeutics in Boston and in Basel. And before entering biotech leadership, Martin has held a series of increasingly senior roles at Hoffmann La Roche, where he's been very famous. A chemist by training with a PhD in theoretical chemistry from the University of Marburg, Martin has spent much of his career at the interface between medicinal and computational chemistry, with a keen interest in knowledge generation and decision making in small molecule drug discovery. Martin is the author of more than 80 publications, co-inventor on more than 20 patents, and has brought multiple compounds into clinical development. The Medicinal Chemist's Guide to Molecular Interactions is, of course, one of Martin's famous contributions to the drug discovery community. Martin is also one of my heroes of medicinal chemistry and one of my early idols. So it's a specific honor to have you here today, Martin. Welcome again.
Well, don't flatter me too much. Also, thank you. Yeah.
No, I think
Yeah.
that's much deserved. Yeah, and in the good old drug hunter tradition, we really like to get to know our speakers a little bit better. So, Martin, can you tell us a little bit about what inspired you to start a career in drug discovery?
Yeah, so as you said, I mean, I started in computational chemistry, and computational chemistry is something that I fell in love with fairly early on during my studies. And the thing that excited me most about it was really this idea of being able to predict something that someone else could then test experimentally in the lab. And I had the luck to have a great partner during my PhD where we could do this already. And then there was no other place really to do this than industry. This is where it all comes down to, whether your predictions are right or not. And so that's how I started at Roche at the time, was really through that computational angle, and then spending more and more time, of course, also with the more practical and wet lab disciplines, but always outside the lab. So I started really from a more conceptual angle, which I think in my case was almost an advantage, because you can think about things in a bit of a somewhat neutral way. But I love that idea of being able to predict and to move forward.
Yeah, no, that's I think, getting increasingly important these days. What's your favorite molecule actually, and why?
So that's always a bit of an unfair question. That's a little bit like asking a mountaineer to pick a favorite mountain out of the many peaks that are there. But I will pick one today, and I'll talk about a little bit about it as well later. And for a particular reason, that's risdiplam. And risdiplam is what I love about it is, first of all, the audacity of saying we can modulate splicing with a small molecule without having initially any proof. So the people at PTC Therapeutics who started this deserve a lot of credit for that. And then the optimization process that I could witness at Roche was also a fascinating story. And now it's out there, and it's a wonderful compound. So I'll pick that one. Although, of course, there are many others where more structure-based input was also driving. And of course, I love the structure-based aspects. In this case, there's zero structure, and the phenotypic aspect of it is even more exciting.
Yeah, drugging RNA is also becoming
Yeah.
increasingly interesting, and lots of cool companies are trying to do that. So really, maybe the next level of biology to drug RNA rather than proteins. So super cool. What are you most excited about in drug discovery today?
Well, witnessing the uh, breadth and depth of small molecule research today is just great. I mean, nowadays we're doing things that we really didn't think were possible. Molecules are getting larger. We're starting to figure out how to deal with these larger compounds, and novel mechanisms of action become possible. So that's really it. I mean, I'm a small molecule person, I'll admit it. So I'm in love with those molecules. They go everywhere. They're wonderful. The mechanisms are so diverse. So that's the expansion of that modality is what I'm excited about.
Couldn't agree more. I'm like you, I'm also very much a small molecule guy too. Well, Martin, if you could go back to the beginning of your career and give yourself some career advice, what would you say to your younger self and maybe, to the younger folks here in the audience that are just starting out their career as medicinal chemists?
Yeah. I mean, like everyone, I started out as a single contributor who had the privilege to learn step by step over time in a larger organization. And I think the learning over time was twofold. One is I learned, you learn more and more about the science and how drug discovery works. And the other thing you learn is how teams work and how teams have to come together to actually get things done. And I think if I going back, I think the advice I would give myself is twofold, really, is on the scientific side is do speak up early enough and question things that are being done a certain way and that don't necessarily need to be done in a certain way just because that was the case in the past. So especially in larger organizations, there are certain ways of doing things. And if you go and say, yeah, wait a minute, why are we doing it this way? That's actually helpful. And so dare to do that early. And the other part is really about you're just paying a lot of attention to how teams click and how teams interact and how to get teams going. Again, that's coming from that single contributor, more scientific side, understanding that that's probably more than half of a success of a molecule or of a team. It's just so important.
Yeah, it's great advice. And I think there's a lot of research these days on how successful teams work together. And yeah, I think the advice that you gave to on essentially like questioning, like bringing a fresh set of eyes to the table, I think is very important as well. Last but not least, Martin, what do you most like about Drug Hunter?
Yeah, and so it's just a great platform for informing yourself about things that you don't know anything about. So I go to it for the target stories, the summaries of how mechanisms work, which companies are active. There are lots of links in there, which is fantastic. It's a little bit like a Reader's Digest for that kind of field, which don't want to sound derogatory at all. The Reader's Digest has a bit of a reputation, but it is a fantastic way of summarizing. But I think what's even more important is that it's got this fantastic potential to become an almost universal knowledge base for that field. And the ability to connect bits and to connect these molecular stories that will also be the topic today is just great.
Awesome. Yeah, thank you so much, Martin. That's greatly appreciated. And yeah, I want to show like before we dive into Martin's presentation, I want to show like a little bit of like what you can do with Drug Hunter. And today I wanted to focus on the structure search, and it'll just be 1 minute. So I already drew like a nice carboxylic acid here. So if you have the question, for example, what drugs recently got approved that contain a carboxylic acid? You can find this out in seconds with Drug Hunter. Just click here on the FDA approved drugs and you can search. You can sort by year here, so earliest to latest. And you can see immediately like what were the most recent drugs, what were their mechanisms, in which context was the carboxylic acid used. And again, if you want to see that for, let's say, clinical candidates, you can do that as well. Now searching the clinical candidate database. So of course, FDA approved drugs, are a little bit older and the discovery stories typically do take a little bit longer. So if you want to see molecules that are currently in development and that are being actively pursued by development organizations that do contain carboxylic acids, you can find those here in the modern clinical candidates as well. So we actually, as you probably know, the INN list just recently released, and we updated the clinical compound database with that information already. Now, if you want to use a carboxylic acid in your own design, I would probably recommend to search the drug hunter molecule database. So now here you get molecules again from the drug hunter database to see what essentially what types of mechanism, what types of context these carboxylic acids can be used in. So for example, here in this integrin inhibitor, you can further narrow this down and you can type in, let's say, integrin and see like excuse me, see the structural differences, see the leverage. And if you're interested in those case studies, for example, you can click here and you will immediately be brought to the case study. And here you can learn about the entire program, or you can also learn how here in this particular case, the carboxylic acid was smartly hidden away in an intramolecular hydrogen bond donor, hydrogen bond acceptor way in order to make the molecule more permeable. So it's a really interesting example for how you can integrate a carboxylic acid into your molecule. And there are many, many more of those interesting stories. And with that, I will stop sharing and I will hand over the floor to Martin for his presentation on molecular storytelling. So, Martin, really excited to hear many more stories about molecules from you today.
So let's dive into this. Let's dive into the wonders of medicinal chemistry together. I'm inviting you on a little walk where we will encounter some history, some philosophy, perhaps a few pointers as to how we can think about things a little bit differently in this fascinating field. So let's imagine you're a medicinal chemist and you have just joined a project that has already been going on for a while. What's the first thing you do? You go to your organization's database that stores chemical structures of the project along with associated data. It may look a little bit like this. And perhaps you look at the first data sheet where you have structures and associated data, and then you do this again and again and again, perhaps a dozen or 100 times. And in this way, you can walk through the entire history of the project. And you can also look at property distributions to understand some trends. And then on the next day, a colleague who's been on the project for a long time comes by and you go to a whiteboard where he or she explains the history of the project. Over time, during that discussion, a picture like this emerges. It's a chronological account of the key compounds that were made and tested, along with the reasons why they were made or the insights that were learned from these compounds. And so by using these two ways of getting familiar with the project data, you now have sort of reinvented two different ways of looking at the history of the project. One of them considers all projects as equal, at least they're stored in a database in the same tabular way. And the other one tells a story made of molecules. Both are useful ways, of course, of looking at the data, but for different purposes. The tabular form is good for data analysis, for looking at trends, for correlations, and it has become a staple. This is how we store molecules in databases, and we publish SAR, often in the form of tables. And like I said, these are two perspectives of the past of a project, and I'd like to name them analysis and storytelling. And we'll deal with the future part in a minute. For the moment, I'll ignore that piece. Let's reflect a little bit on this dichotomy, analysis versus storytelling. As a community, we've worked hard to try and rationalize medicinal chemistry, and if we're focused on the analysis side of things. And I claim that the storytelling approach deserves a little more attention because stories are so extremely well suited for learning and for understanding specifically in this discipline and ultimately for getting better at it. The generic version of a storytelling tree looks somewhat like this. There are roots at the bottom. They could be screening hits. They could be compounds coming from a completely different project. And then There's SAR development along these branches, and some of those branches can then come together because you realize that you can make hybrid molecules. There's SAR transfer. There are, apologies, there are terminated branches, and then there are clinical candidates that move forward into development. And so frequently, when we think about the history of a target, these trees exist somewhere, but they hardly ever come together visually in one place. So we may see a partial tree published as a review article. And here's an example. This is an old one from 1994. So this is from the Glaxo review of their angiotensin 2 program that shows how their chemical series has evolved. This is an upside down tree, so it starts at the top, goes to the bottom, where you see how SAR has diverged, and then compounds have come back together to form hybrid molecules until you have the final lead. So you have to search quite hard to find these kinds of depictions in the literature. But the point I'm making is they really work well with us. They have this immediate effect of recognition, of understanding. And in my career, I've often asked medicinal chemists to tell me the story of their projects, and the outcome has often been this type of graph. So let's tell more of those stories in this forum. That's one of the things I'd like to say to the community here today. Now, often, of course, what's essential is to understand where do these trees start. Here's an example from NLRP3, so the compound here in the center, which was also recently featured in a talk here on Drug Hunter, was the starting point for many endeavors, many starting points for many biotech companies who then turned this compound into developable compounds, which are now in the clinics. And you can see how close they are structurally. And so in a way, the trees that we find and that we can that we can compile retrospectively don't always simply tell a story of science. They also tell a story of commercial interest and of overall field interest in a specific target. And in this particular case, it took quite a long time until Novartis then came along and said, we have run a high throughput screen on the same target and came up with a lead molecule that's depicted here on the right-hand side, which then in turn led to many, many clinical candidates by other companies. And I'm not even depicting half of these right here. In this particular case, the two trees stay separate, and they stay separate because in the binding site, they do overlap, but they hardly overlap. They have very different binding modes, and so making hybrid molecules between those two becomes hard, and therefore the trees stay separate. But the point here is that the starting points, these root molecules, play a particularly important role in our field. So oftentimes, the unsung heroes in medicinal chemistry are those who take a high throughput screening hit list and then do the serious detective work of figuring out which compounds need to be followed up or should be followed up, where are the true hits, where are the false positives, until you arrive at a starting point that's really then worthy of becoming the root of a molecular story. And often the structural changes along the tree branches are not large. So here are a few examples, and I'll walk through them to make a few additional points about these trees. So going from left to right, I'm often essentially telling not just a story of an entire tree, but really just a pair of compounds here. The first example is from a Roche cathepsin S inhibitor project, where a single nitrogen to carbon change led to a dramatic jump in potency, and in this case caused by a change in conformational preference. And we've, of course, become used to call a pair of molecules like this with only a single structural change, a matched molecular pair. And it's interesting to think about matched molecular pairs as the place where the data analysis and the storytelling meet. A matched molecular pair is the smallest unit of molecular storytelling. You go from A to B, and then if applied to molecular properties and to many of the same matched pairs, you can derive transferable rules. The key is that these rules are often so extremely contextual that generalizations easily blur the picture. But we'll get back to that later. The second pair here is about a change in selectivity. This is moving from an unselective beta-adrenergic antagonist to a cardioselective, that is beta-1 selective one. And the clinical impact of this discovery was enormous. So we'll keep that in mind. The observations we make as we grow these trees are not necessarily always about optimization, about better properties, about moving towards a candidate, but also about really moving into new biology. And the third pair shows how the large tree of diazepam compounds that has originally yielded so many GABA A receptor modulators has branched out to give us the first widely characterized bromodamine binders from the Bradner lab. So here, one molecular tree has spawned a new one, and that's exactly what we are attempting to do every single time we screen our historical collections against a new target. So we actually hope that this happens a lot. Now let's think a bit more about this tree nomenclature. So I've called these stories trees. And this, of course, sort of begs the analogy to an evolutionary tree. Here, this is the pedigree of man. We all think of Darwin or other people who have come up with concepts like these. And if you call it a tree, then you imply a sense of hierarchy. There's something at the top here. In this case, it's man, or it's our clinical candidate, that is the most valuable compound. And that is sort of the culmination of the years of work. And of course, what we're trying to do as medicinal chemists is, in fact, a form of directed evolution. But if we focus too much on directionality in our storytelling, then we lose information on what seems like dead ends, but can teach us a lot. Information on failed projects or on unproductive strands of the SAR development is what I would call the dark matter of medicinal chemistry. It's not visible, it's often not poked about, but it's incredibly important because dead ends are usually the moments in projects where we've learned something. We've learned something about biology, we've learned something about our target product or target compound profile that we didn't know about before. And the funny thing is that the fewer molecules we use to tell our story, the less space there will be left for a dead end and for these dead end branches that teach us so much. So we tend to be quite selective in hindsight. And so this question of how many molecules does it take to tell a story is an interesting one, right? Because you could say, if you swamp the field, if you share every single compound that you've made, you've blurred the picture and you lose the story. But if you take too few molecules, then very little is left about the wandering to the left and to the right of the path that was ultimately successful. So it's definitely a Goldilocks problem. You can be right, sort of right about in the middle. But it's surprising to me that it doesn't take too many molecules to actually tell the full story of a project in the end. And so, if you summarize a project that maybe has generated something like 1,000 or 500 molecules over the generation of years, usually about 20, maybe 30 will be sufficient to give you the cornerstones of the thinking of a project. So I'd like to just encourage you, as you're listening, to think about that for yourself. How many molecules do you need for telling your own stories? So I've looked at this tree for long enough. Darwin himself, when he was thinking about evolution towards the end of his life, was actually saying, well, actually, the tree is not a good idea. we should be talking about a coral. Other philosophers have said, let's use a rhizome, this root cell, the set of roots that, for example, bamboo grows out of, as a model for how knowledge grows. And that, I think, is the much more fitting picture because of a rhizome or a coral, they will grow in any direction where there is space. And that's often what we do in medicinal chemistry as well. We don't know from the beginning where the successful branches will uh, really be. You get the idea. Let's not get bogged down in botanics here. Um, but such diagrams are really not unique to medicinal chemistry. If you're in epistemology, so in the philosophy of how knowledge is generated, then much of the evolution of science in general can be depicted like this. And here's an example of what the philosopher Stephen Toulmin has done in his perspective of knowledge evolution too. There are new ideas that develop that are more or less successful. They get recycled, they get abandoned, or they get adopted by the community. And so these trees, again, are something very general in knowledge generation in many, many, many fields. The nice thing about medicinal chemistry is that we have something in hand to tell the story because the compounds we have are actual physical samples. They can be cornerstones of a story, and very few other disciplines have something like that. So I left you with half a diagram when we were talking about the past of projects. And now it's time to look at the future as well. So the corresponding future aspects of analysis and storytelling, in my eyes, are creation and prediction. I think the prediction part is easy to explain, right? Because as you go through an analysis of, for example, log-D values or solubilities or permeabilities, you can build models from many measured data points and then turn that into prediction tools. The storytelling piece turns into creation as you go into the future. That's the key thing about medicinal chemistry, is that it has to somehow learn how to integrate all the data that's relevant about a particular compound and then turn it into a recipe for making the next molecule in a reasonably targeted way. And to just illustrate that a little bit more, I'm going to use a single example today, and that is essentially the final chapter in the optimization of a molecular series towards risdiplam, we mentioned at the beginning. So we're jumping right in. Here's a compound that was already quite advanced. It does what it's supposed to do. So in other words, it does induce a change in SMN2 splicing. You see that in the green chart. It's also active on an off-target on FOXM1 splicing. That's part of the mechanism. This is something that cannot be entirely separated from the SMN2 activity. But in this particular case, there was a metabolite of the compound, the N-dealkylated, demethylated metabolite that stayed in the periphery, was not brain penetrant, and therefore only contributed to the side effect and not to efficacy. And so that was not desired. Also, the compound had a phospholipidosis risk. And so the goal was to reduce the volume of distribution of the series. So now you have a situation where you have two things that you want to do, two macroscopic observables that you want to change. And the goal is to translate those into structural changes. And so that's really where I think the key is to medicinal chemistry, creativity in general. You want to achieve something, you want to change the property profile of a series or class of compounds. And you have to take that data that is being measured and turn it into a recipe for structural changes. And that's a creative, analytical, combined process that requires both these prediction elements and the future-oriented storytelling, the creation. So if you take problem number one, in other words, the identification of a terminal cyclic amine that does not have a dealkylation risk, that's reasonably easy to translate into a structural change. You just, either find cyclic amines or you have amines that do not have that N-alkyl residue. The second one, reduction of volume of distribution, isn't that easy to translate directly into chemistry. You first have to think, okay, what is it that affects my volume of distribution, then translate that into chemistry. And in this particular case, it was known that the series had a strong pKa dependence for volume of distribution. So you see the compound that we talked about has a very high volume of distribution, and there's almost linear correlation within a certain pKa range, where if you decrease the pKa value, you get lower volume of distribution. So that was then taken as the recipe for changing the chemical structures in this particular case. Also, it was clear that a certain minimal pKa was required for retaining efficacy. And so now we have a pKa window within which we have to operate. And so turning the macroscopic observables into structural changes is what led to this overall recipe here. No dealkylation, keep the log D in a certain range, and pKa between 7 and 8.5. And so at that stage, you can take that series and say, all right, what do I replace this amine with? And you can go back into your databases and look for amines that fulfill these properties. And there's a desired range within which you can operate. Many of those were tested. And the one at the very bottom here on the left-hand side was, in fact, the winner. This is the lowest possible pKa that reduces volume of distribution, therefore the phospholipidosis risk, but keeps the compound very active. And so in going through that set of transformations now, you, as a chemist, in this case it was Hassan Ratni, a former colleague of mine who led that project, really went through this exercise where initially there's, theoretically at least, an abundance of choices of things you could do with these compounds, and went then ahead and said, for these and these reasons, I'm going to reduce that abundance of choices to this set, and here's the reason why. And that, I think, is what, in the end, the creative / analytic predictive combination of medicinal chemistry is all about in the end. And what's fascinating is that along this way, tacit knowledge, of course, still plays an enormous role. When different chemists looked at that problem, they would come up with slightly different perspectives of what to do because of the past projects that they had seen. Whatever you have seen in the past will pop up again in your mind and say, well, maybe I can try that again. And I think the future that we all would like for our field is a future where the little learning nuggets that you can collect over the years and years and years of medicinal chemistry, they sort of pop up somewhere, either on your screen or in your head, directly associated to the type of problem you're currently working on. And I think there are systems that could be built that are associated to that storytelling concept that would really support that kind of thinking process and that kind of lookup process really well. So that's the end of my SMN2 story. But there are, of course, many, many more stories. And I'll just show one more example that I think is particularly worthwhile talking about, in particular in this time and age of high molecular weight compounds. So this is one figure from a publication of the Novartis Werner helicase inhibitor program. You can see these are quite sizable structures. By the way, I think one of those great stories about how to find out which of your high throughput screening hits is a true hit. And I've highlighted two tiny little fragments here in these molecules. And these are cases where it was all about introducing the right kind of polarity distribution in the molecules in order to both keep lipophilicity reasonably low, so solubility reasonably high, and keep membrane permeability high at the same time. And so those are why we're talking not directly about matched pairs here along this particular graph. Those are the kinds of introductions of tiny little changes in a molecule that are extremely worthwhile memorizing because they will be transferable to other situations where the same kinds of problems are being solved. And so Henrik Möbitz has a flash talk on this platform that's highly useful in that context and that I really recommend to everyone. Those are the kinds of things that we need more of. So far, I've shown you molecular stories that look like this. So there's a molecule, there's something we learn, leads to a next molecule, and so on and on. And that's how, when we think about optimizing compounds, we will typically depict our stories. And the more we are in an area where we know what the assays are, where we know the biological problem, then that's probably a useful way of depicting it. But the inverse is also possible. You can also put the compounds on the edges of that graph and the insides, place the insides as the nodes. So essentially then one molecule leads you to an insight, and then by means of a next molecule, you arrive at the next insight. So essentially you're inverting the molecular graph here. And I think this is really how medicinal chemistry came to pass in the first place. If you go back 100 years or even a lot less, then molecules have been entities that have allowed us to understand biology better. And then, of course, we're no longer in a pure optimization setting, then we're in a setting of understanding biology better. And I'm just going to give you a few examples. These are all historical ones that should remind us of where pharmacology and medicinal chemistry really come from. When people started working with nicotinic receptor antagonists for the first time, then that helped us derive the concepts of occupancy and affinity and efficacy. And then radiolabeled assays helped purification, sequencing, and then, of course, ultimately cloning of beta adrenoreceptors that I had mentioned before. And then the selective adrenoreceptor ligands helped us to separate the subtypes in tissue assays and so on. And there's an example further below here where various different selective small molecule TLR modulators have helped us understand pathway-specific innate immune responses. All this was with the help of small molecules. And so a small molecule is not just an entity you need to optimize, it's also an entity that allows us to learn. It's a tool. It's essentially like a scalpel that allows us to cut into bits of biology. And we need to remember that we don't really understand much of human biology. So when we think about AI-optimized drug discovery today, a tiny bit of that kind of humility is helpful. Here's another example. I just think it's great to remember these kinds of stories. So at the bottom here, you see a compound from the 1930s called Prontosil, which comes directly from the dye industry. And it had a metabolite called sulfanilamide here in the middle. People learned and relatively quickly that this was an inhibitor of a recently discovered enzyme called carbonic anhydrase. And now sulfanilamide was the basis for many, many additional compounds. They are all in this sulfonamide, sulfonyl urea space, where new discoveries were made essentially almost every year and then later every decade, up to the point that a compound like carbutamide essentially named its own receptor. So the sulfonyl urea receptor that leads to these, that has led to these hypoglycemic drugs here, was named after the compound. So again, compound allows biological discoveries. So, while we're no longer in this purely phenotypic phase of drug discovery, oftentimes, of course, phenotypic approaches do still have an enormous value. If you can't take the molecular mechanism apart right from the start, then phenotypic approaches still work wonders, and we discover new biology in this way. And I think even today on Drug Hunter, you see these examples on a daily basis. If you have a system that's as complex, for example, as glucocerebrosidase as an enzyme. It does require small molecules to understand how to drug this over time and understand biology better. So we will always be with one foot in the exploration and with another foot in the exploitation phase. When all the assays are known, when it's clear what we want, when there's maybe already a compound out there that fulfills the kind of profile that we think we need, then we're fully in exploitation space or in optimization space. But most of the time, some of what we do is exploration. And here, I think the language that we use as chemists becomes important. So if you picture yourself as a medicinal chemist who optimizes any kind of series of molecules, whether you change the phenyl ring and substitute it by a heterocycle at the beginning of a project or at the end of the project may sound like the exact same activity to an outside listener. But you do it in a very different information context when you do it in an exploratory fashion, just to see what the SAR looks like, or when you're trying to fine-tune the last bits of PK before you nominate a candidate. And I think what's important is that as chemists, we start to bring that context into our optimization language. So it sounds no longer exactly the same by saying, I'm modifying the southern phenyl ring here. But by starting to describe the information richness of the landscape and then saying, and within this context, I'm exploring this phenyl ring substitution, for example. So it really boils down to how we tell our stories and how we bring that information context into our storytelling. Well, with this, I think I'm already at the end of what I have to say. Medicinal chemistry to me is a storytelling discipline. This is how we remember, this is how we learn. And we might want to adapt some of the tools and the language that we use. I think visualization tools that help us to build these trees are important. Database structures that allow us to connect the stories across different companies, across targets might be helpful. And then finally, annotating these little arrows between the molecules with the kinds of insights that could be transferable is just, I think, a great goal for us to have as a community. And I'm just looking forward to more discussion about how we can achieve this together. And so while I started with an AI-generated picture with some botched molecules in it, I'm going to end with a picture of alchemy, just to remind us of our roots, that there's still a lot to be discovered out there, and that there are a lot of wonders of biology that we can explore with our molecular scalpels. Thank you.
Awesome. Thank you so much, Martin. That was very, very insightful. I mean, yeah, I'm definitely going to echo you here. It's so important to have those insights at your fingertips, that knowledge that you're looking for when you need it. And that's oftentimes really, really difficult to find. And that's obviously one of the things we're trying to solve here at Drug Hunter. So yeah, with that, we have a number of questions already in the audience, starting here with Thomas Babinski's questions. You were talking about the questions, how many molecules do you need to tell a good story? So Thomas is wondering about, what makes a molecule worthy to be included in a story? And what's your answer to this question of how many molecules do you need to tell a story?
Yeah. Well, I think that's hard to answer in a very generic way. I would say that there has to be some sort of insight associated with it. So you might explore one particular region of a molecule quite intensively and modify substituents as much in a very broad way. And then you'll start to realize that one particular one of those maybe really changes something, and that could have something to do with affinity or with selectivity or with something else. But um, those aha moments that you have when you look at your sort of biweekly report from the assays, those are the ones that I think lead to those cornerstone molecules in the story later.
Yeah, sounds good. Well, next question is from Niels Westkamp. So he says, very nice talk, Martin. And he says, the thought process that you describe with matched pairs and congeneric series, that typically assumes additivity of properties and
Yeah,
requires that the binding mode remains
yeah,
stable.
yeah, yeah.
So,
Yeah.
right?
That,
He's
that that's a
wondering.
great additions, and it's something that I should have spent a little bit more time on. It is a property of our minds, I think, that we have to live with a somewhat reductionist view and a somewhat additive view of SAR until it doesn't work anymore. And when it doesn't work, then I think we know. So then we either get the kinds of synergies, the kinds of multiplications of affinities that we essentially all rely on in a way in medicinal chemistry when we optimize compounds, or we get the opposite. We get a frustrated system. And both can happen, of course. So yes, it's important to keep in mind that additivity is not the answer. And it really also depends on what we're looking at. So if this is certainly true for SAR, in other words, for everything that's sort of affinity based. Um, but for properties, the rules may be different. There are also going to be cliffs, right? When you have a sudden conformational change, for example, and not everything can be explained just by looking at the 2D structure. By the way, also something that Henrik Möbitz really points out a lot. But there are more sort of areas of continuous optimization, sort of trajectories that you can apply when you look at properties like log D, for example.
Yeah. And yeah, Niels was wondering, like, how would you, like, in kind of the molecular storytelling, capture those kind of surprises that don't add up? Is there any like, advice you would have for folks?
So that's also a good one. So what I've done here is simply shown these trees or corals or whatever you call them,
Hmm.
and that's maybe only one way of depicting the progression of molecules. There will always be an element of we go from A to B to C, but whether it's a tree or not, or whether there are maybe different ways of visualizing these, that I think needs to be explored with the kinds of in the kinds of interactions with software developers and visualization geeks that we now have. So I think that's a journey we are still on.
Sounds good. Well, next question is from Anna Sternberg. She's wondering about the Werner helicase inhibitor story that you told with the assay artifacts. So, what are the indicators whether a hit in high throughput screening is real or an artifact?
Oops. Yeah. Well, so I mean, the first thing is, of course, to understand that when you've run a high throughput screen, then by definition you look at that end of the distribution where the artifacts are. So that's there will be artifacts always. And so I think it's really about how do you design orthogonal assays that help you tease out what a molecule does from multiple different perspectives. But then also to have and this is more, I think, a gut feeling kind of thing, this understanding of when is it time to actually start chemistry around a compound, even though it might still be a very weak one. And to say, this one looked clean, it's weak, but now if we see a bit of SAR emerging around it, it may be a starting point. And I think this is how the starting points that led to risdiplam later were actually found. They were very weak compounds. This is how the Werner compounds were identified. At some point, you have to say, yep, this one deserves some chemistry now.
Yeah, for Werner, I think I recall there it was a singleton hit, and it was really important to, as you pointed out, have like a cascade of assays set up to really validate the hit downstream. So yeah, super, super important.
Yeah.
Um, Pavel is wondering, what are the best practices for storytelling when it comes to persuading medicinal chemists to use result from computational approaches? You may have many stories there, Martin. So he is wondering
Yeah,
about a
yeah,
predict first approach. Yeah.
that's a nice one. So I think nowadays that's actually a little easier than I think it was 20 years ago or so. You can simply say, you want to be somewhat rational in what you do. And so oftentimes it's helpful for project teams to have a person, and that could very well be a computational chemist who is the one who helps rationalize why a certain compound needs to be made. And it could be that the computational chemist says, we have to explore the pocket in this way or in that way because that's how the conformations work, how the interactions work. And then say, and at the same time, we're going to challenge the model as well. And so we'll try consciously a few things that go against the model just to see how it holds. I mean, if you have a really high resolution crystal structure, there isn't much to argue with it. But if it's a model, if it's a whatever homology model based or something else, then of course there's some uncertainty to it or to the binding mode. And then you should also go in and say, let's challenge the model and let's do that together. Um, but the the dialogues between the um, medicinal chemists and computational chemists, that's always going to be an interesting topic. Which I've always loved these interactions. You just have to engage in them.
Nice. Well, on the other hand, Doug Williamsons is wondering, quite often we need to engage stakeholders or decision makers who are non-chemists and
Yeah.
who are not structurally aware. So, what are your tactics or suggestions to engage those in those types of discussions and future plans?
Yeah. So uh, sometimes, we've been spending the last hour in this world of medicinal chemistry, where, of course, you can show structures, and structures are a language that is being understood. And it's clear that that's not always the case. It does help sometimes to, simplify things a little bit and to say, I've explored a space that is very, very dense here, and I've moved out of it, and now I'm in a completely different property space, and then just take elements of that structure and say, this is the bit that's now different, and this is what it does to the compound, to be a little bit more educational at the same time. And so sometimes it's helpful to just explain bits of chemistry, but only bits of it, not the entire chemistry textbook. Um, and to work with, I think to work with this concept of information density has been helpful for me, at least, to say, here I know exactly what I'm doing because I'm purely optimizing. Here I don't. And to say, this is where we need to explore and expand a bit. Here's, it becomes a little more a question of just seeing what we can do with a fishing expedition.
Yeah,
Yeah.
makes total sense. Yeah. Well, the other thing is you spoke about the importance of dead ends, right? So,
Yeah.
with that respect, Yoshi is wondering, could you comment on how to bring together these frequently opposing forces? So, A is among the drug discovery community, we should promote storytelling and help advance our field. But two, companies generally don't want other companies to learn from their in-house experience. So there those two
Yeah.
forces that,
Yeah.
battle each other. How can we bring those together?
Yeah. It's not easy. What's at least within larger organizations, and I've had the luck to be in situations like this, is to make sure that you don't forget your own stories and to bring them back for publication later when they're no longer as hot as they were. Just publish later, but make sure that you have that material ready if you're interested in publishing. Um, or, submit batches of data to Campbell and just enrich the public data sources in that way. That, of course, is a much lower hurdle in many cases than writing a full paper. Uh, but you have to have the energy for it. And, if you don't have the incentives in yourself, then you won't do it.
Yeah, that makes total sense. Oh, Gagan is wondering, in the age of AI in drug discovery, how can a medicinal chemist play the role of an explorer? And is he allowed, or are they allowed to be an explorer in the age of AI?
Oh, yes, absolutely. I think it's just the AI, whatever is meant by AI in this context, is really is a tool, and the decisions are still with us. And I think again, it goes back to this question: how much do we really know about the problem we are trying to solve? Is it in fact an optimization problem where all the tools and assays and parameters are fixed, or is it not? And that's that I think defines very much whether we're in exploration or in exploitation mode.
Yeah, that makes total sense. Well, Caitlin is wondering about how do you feel about the field of multi-omics and the need for storytelling there? The wet and dry lab continuity mixed into the various ways of looking at biology. Is anyone out there doing a good job of telling those stories right now?
Um, so that is, yeah, again, very context dependent. I think the dealing with multidimensional data, um, the question is, what do you want to get out of it ultimately? So, you have that, whatever that is, multi-omics data set, and then what's the direction? Why have you compiled it in the first place? I guess is what I would ask, and what do you do with it there? So and often this comes in phases. So in my previous company at LifeMind, we had enormously large data sets. We had 100,000 fungal genomes, and then we're mining for natural products in those genomes. And there's this entire phase where the data just explodes. You get expression data, you get RNA-seq, you get this and that. And it's only there for the purpose of finding that compound. But once you have it, that's the distillate. That's what you deal with. And then you move on and do other things. Then you move into regular drug discovery. And so I think that, all the omics data sets, they have their value in a certain period of your discovery process, and then they've served their purpose.
Yeah, it makes total sense. We have another kind of maybe a comment and then a question. So during a project, the story can be smooth sometimes and rough some other times. But the most important part is to take the right decision and pivot very quickly. So, what
Yeah.
is the best way to capture stories for projects when scientists only want to share success stories? Like the incentives are, like success stories are kind of yeah rewarded, where it's more, I guess,
Yeah.
like how do we reward more sharing of dead ends and reason, maybe exploring why those ways haven't worked out.
Well, the success in a dead end is that you've learned something.
Exactly.
So focus on that. That's my answer. Yeah.
How
So
can
it's
we
it's
share
about
this with
learning.
others, right?
Well, if there's something in there that others can take home and say, I know what they've done. They haven't reached maybe the initial goal, but they've learned something really valuable, and now the project looks different. That's fantastic. So it's always this, it's not a failure. It's a learning step. Um, but I guess that's also a cultural question in the end.
Yeah, that makes sense. Well, I guess we're at the top of the hour. Maybe one more last question I want to ask you is also about kind of large language models. So it's from Govindan Subramanian. He says, "Great talk. what is the best way to capture the insights from different projects into a knowledge base and use this information for querying for a handicap we observe in a new project or for like if kind of a problem.
Yeah.
Um, I
Yeah.
do not seem to see a consolidated path that allows us to allow such queries like in LLMs. Do you have any thoughts on this?
Mhm. Well, so in my mind, maybe I'm a little bit old-fashioned there, but I go back to this concept of a matched pair, which is a transformation. And so if you encode learning steps as transformations, that's a way of storing them because then they can be matched onto um, a compound or a pair of compounds that you find later. You can say, if either one of those elements is found in your target structure that you're currently working on, then think of this because other people have been in that situation before. So it's more about in my eyes as a sort of cheminformatics person, you sort of encode the smarts for the two end situations and then use that to match onto whatever your database of molecules is.
I think
That's one
that's
way of
good.
thinking about it, but there are probably other ways that others can think of. Yeah.
I think that's good advice. Well, we're at the top of the hour. Thank you again, Martin, for a really phenomenal talk. We want to thank the audience, of course, as well, for being super engaged today. Lots of questions. Unfortunately, we didn't have time to answer all questions. There are a couple of more questions, but
Yeah.
um, that's really much appreciated. And yeah, with that,
Yeah.
we will. Yeah, Martin.
Also, from my side, just wanted to say thank you for all the questions and for listening. This was great.
Amazing, amazing. Yeah, so we will be back here next week. Don't miss this. So we'll talk about the discovery of zolucatetide, also known as FOC001. It's the beta-catenin TCF4 protein-protein interaction inhibitor. So really exciting story next week. Don't forget to dial in. And until then, we wish you a wonderful rest of your day and a wonderful rest of your week. And thanks again, Martin, for the great talk. All right.
Bye bye.
Bye now.